Hello and welcome to the PyMINEr tutorial where I'll walk you through what the results look like. So this is the summary page that is found in this PyMINEr summary.html file, where you can just open it up in Firefox or whatever your browser of choice is, and this is what it should look like. So the first thing at the top that you'll see is the annotations file, and this is something that's generated by PyMINEr's integrated functionality with gprofiler, where it will take your gene IDs that are in the leftmost column, and here we were using mouse ensemble genes. So this is the initial gene that was in your original file, and it'll convert it to, if it wasn't already an ensemble ID, it'll convert it to ensemble IDs, give you the gene symbol and the description. And so that's the basics of the annotation file. And if you are not using a data set that is human, it'll also automatically figure out the human orthologs. This is important for PyMINEr's functionality for generating the autocrine and paracrine signaling, but it'll also be useful if you just want to know what the human orthologs are. And this is what that file looks like, where you have your initial gene IDs, and then it gets mapped over to the ensemble genes, and then the human ortholog to that ensemble gene, and then again the symbol and the description. Okay, the next thing that you'll find is some plots that are made by the clustering algorithm. So the clustering in PyMINEr by default uses over-dispersed genes contained in your data set and uses a z-score cutoff of 0.5 for the residuals of the locally fit curve of the regression, using a lowest regression model of the over-dispersion plot, which is the variance versus the mean expression. But that's a little bit in the nitty-gritty. If you're not into clustering, you don't have to necessarily worry about it. But in one of the future tutorials, I'll get into how you can supply your own sample clusters if you want to use a different algorithm. But overall, this file gives you the annotations where you have your cell here and the group number here. So in the right column, you'll have just a simple integer, 0, 1, 2, 3, etc. And if two different cells have the same number, they're a part of the same group, or cell type for a single cell RNA-seq. And so here is the object that is actually clustered in the default parameters that are used for the clustering algorithm. And what this is actually an affinity matrix based off of the Spearman correlation matrix. So the darker the color here, the more similar the cell types are, the different cells are to each other. And the colors along the sides here indicate whether or not that block of cells or samples is a part of the same group, or in this case of single cell RNA-seq, the same cell type. And then here's a simple PCA projection, and it's also color-coded by the sample group here. And the key, the legend, is over here to the right. And so this is a random sample of the genes that were used for clustering. In the default settings, this is going to be the over-dispersed genes. And this is, again, just a different view of the affinity matrix, where they're clustered by cell type here, and then unsupervised hierarchical clustering on the other axis. And this is without any of the supervised clustering, with just the hierarchical clustering on both sides. And then this is the same thing, but with the colors along the side indicating the cell types. And final is the t-SNE projection. But something that you'll notice is that oftentimes if you have a small number of cells, you don't see really great separation using t-SNE projection. So those are the clustering plots, and here we have some basic statistics where we have group level means and also the standard deviations. So this is useful if you want to make a bar chart or something like that. You can just go over here and look at this variable, this gene here, and then in each of the columns here, you have the mean expression of that gene. And I'll note that this is a text file, but you can easily open this up, or really any of these text files. If you can open this up in Excel if you just click this, and then once you've finished that, can open it up, and then just drag it over to Excel or whatever program you're using, and it should open up just fine like this. And so another thing that's in there is the group level z-scores, and then the last thing that's in there is the Benjamini-Hochberg corrected one-way ANOVA, is looking for whether or not something is differentially expressed across all of your cell types or groups if it's not single cell RNA-seq. So this tab, Gene Enrichment in Groups, contains a list where it'll have a table of true and false, and what that means is you have, let me open it up first so you can see what I'm talking about. Bear with me a second here. So again, this is this true false significantly enriched table, and if you want to see what genes are pretty high in each of your sample groups to possibly help you figure out what your what cell types this actually is, you can go here, and now you have a list of genes that are relatively high in sample group 0. So true means that it's relatively high, and false means that it's not enriched in that cell type or sample group if you're not doing single cell RNA-seq. And another part of PyMINEr that's fully integrated is pathway enrichment on each of these enrichment lists, and you can look at them here, where you can look at them individually and look at which pathways are enriched in each of the different cell types. And so here in sample group 2, if we look at the top result, it's psyllium, and if you're very familiar with the airway, you can probably guess that this cell type now is the ciliated cells, which help to brush out all of the mucus and garbage that we inhale all day. And here we can see the combined pathway analysis, where it just collates the negative log 10 p-value of each of the different pathways that are enriched in all of the different groups. And here again you can see psyllium, which is ranked very high as being important by PyMINEr for being likely a contributing pathway to the differences between your different cell types. And another really useful aspect of PyMINEr that is good for trying to figure out which genes are, or which cell type each of your groups is the highly expressed marker genes. And what this is it looks at which genes seem to be relatively exclusively expressed in a single cell type. So to be counted in this group, you have to be significant by one way ANOVA, and we also look at the distance between the highest expressed, the highest mean expression in each group and the second highest. So we want this to be pretty big, where the cell type that's expressing the most of this gene is leagues ahead of the second highest expression level in the next cell type down. And then we also use the Q-value metric, which is used to identify outliers in some cases. And this is just a ratio of the thing that I was just talking about, that metric where you look at how big is the gap between the highest expression level and second highest, and then it's the ratio of that number to the full range, so highest versus lowest. And if all criteria are met, then it makes this list. And here's a heat map of all of the genes that made the cut there. And so if we open this one up, it looks like this. And so this file will help you identify cell type marker genes. So if you know what cell types you're expecting, you can go through this file and look for known marker genes. And if you don't know what cell types you're looking for, this will allow you to identify genes that would make good candidates for later bench validation. So the first thing that you'll probably want to do with this file is to sort it based off of the distance from the max to the second max. So let's sort based on that column. And here at the top, we're going to have the best cell type marker genes. So if we first want to go and look at each cell type on their own, let's filter this last column, which tells us which cell type it's enriched in, and look at sample group 0. So sample group 0 has the GABA receptor, MUC1, cytochrome P450, and here's another cytochrome P450. So this is most likely a club cell that is somewhat inflamed, because it's been shown that club cells in an inflammatory context can start to induce expression of mucins. So let's have a look at the next cell type on the list. Sample group 1. So here we have glycoprotein 2, which is a granule membrane. What else pops out? MUC5B. So these are going to be the goblet cells that are typically the cells that express mucins, and this is a more canonical mucin that's expressed in the airway. So what about sample group 2? And top gene is alpha-tubulin, so we can be pretty sure that this is the ciliated cells, which have a very high level of cilium, not just the primary cilium, but have a bunch of cilia at the head of the cells on the apical side. And there is another transcription factor that's highly enriched in ciliated cells, FOXJ1. Let's see, yes, there it is. So we can be pretty confident that this cell type, sample group 2, is the ciliated cells. So last cell type in this data set, sample group 3. Let's see what we have. Keratin 5, 17, and 14. So the keratins are very highly expressed in basal cells, which are the progenitor for the airway. And here is basal cell adhesion marker, adhesion molecule, which even has basal cell in the name of the gene. So we can again be pretty confident that this group is the basal cells. So that's the long and short of how you can use this file to figure out what your cell types are if you have known markers in mind before you even do the run. So back over to the PyMINEr results, and then now we can look at co-expression graphs, another interesting aspect of PyMINEr. So what we're looking at here are the co-expression graphs. So on this graph, each point is an individual gene, and what I'm showing here is, or what PyMINEr shows here, is the overall, what some people call the hairball view of a co-expression graph, where each point is a gene, and how close they are together means that those genes are very tightly co-regulated. So they are expressed together in a concomitant way, where if you have high levels of gene A, you also have high levels of gene B. And what that'll look like here is this would be gene A, and over here would be gene B. So all of the genes that are localized in this general area are highly co-regulated. And what you can do is look at your adjacency list. So you can open this up, and what this is, it's whether or not your two genes that are in columns A and B, as you'll see in a second here, if these two genes are significantly co-regulated by Spearman correlation, which is noted over here, then they will be in this list right next to each other. And so if you have a gene of interest that you want to look at, then you'll also be able to see whether or not you'll be able to make a list of how close each of your genes in the genome is away from your gene of interest. Now, if you know a priori a couple genes of interest, that's another aspect of PyMINEr, which I haven't included yet in this tutorial, but I'm going to make another tutorial which will show you that output. If you already have some genes of interest in mind or cell type markers that you want to use, it'll automatically make heat maps of that, etc. And it'll also tell you how far away each gene in the genome is in this cluster map from your gene of interest. And lastly, for the co-expression graphs, we have some graphics made just by overlaying the z-score enrichment for each group. And so red here means that those genes are highly enriched in that group. So here, sample group 1, this hub is highly enriched for sample group 1. Sample group 2, which was the ciliated cells, is highly enriched in these two hubs over here. And sample group 3 is highly enriched for this area of the graph there. So the last thing that I have in this aspect of PyMINEr's tutorial run is the autocrine and paracrine signaling. So there are a lot of files in this, but the first summary file that you can look at is just this this table here, where you have sample group 0 signaling to sample group 0. This is the autocrine signaling within sample group 0. And there were 28 genes that popped up as having an autocrine interaction where sample group 0 secretes a ligand and also is expressing the receptor for that ligand. And there was actually the same number of interactions with sample group 0 and 1, and etc. This is just really a table to give you a rough guess, or a rough high-level view of how big of an interaction there is between each of your different cell types. And then here we have a detailed list of each of the different autocrine and paracrine signaling mechanisms. And so if we go back over to our downloads folder, where I just put this autocrine and paracrine signaling network, and open it up in Excel. Here on the first column, we have the annotation of whether or not an interaction is autocrine or paracrine, and then cell type 1 and gene 1 is just the annotation for each gene. What cell is this gene coming from? And this is the gene symbol, and it tells you where is gene 1 localized, and then does it signal to the same cell type. In the autocrine case here, we have sample group 2, which again was the ciliated cell, is signaling to itself with two extracellular localized genes, SPP1 and FN1. I don't know what these are off the top of my head, but each of these is going to be annotated. So for example, again, sample group 2 and sample group 2, two different collagens are secreted by the ciliated cell in this data set, and those collagens, once they are extracellular, will interact with each other. And to get a better sense for what's going on at a large scale, we have an annotation of which pathways are signaling between each of the groups. So we'll have a look at this one and save that, and then open this up again in Excel. So open this up, and so here we have sample group 0 signaling to sample group 0, which I believe, if I remember right, was the serous cells. Maybe, I don't remember. But sample group 0 is signaling, it appears to be signaling through S100 a lot. Also proteoglycans, calcium dependent protein binding, etc. And if you're interested in a specific pathway, then you can just scroll down and see what tickles your fancy in your given system, whatever it may be, and see what pathways are enriched with the signaling between each of the different cell types here. And so this is a table of all of the negative log 10 p-values for each of these pathways. And what that looks like is this, where we have first the PyMINEr importance metric. So this is again based off of information theory and entropy, more specifically KL divergence. If you don't know what that means, not important. Basically, the higher it ranks, the more interesting this pathway might be for you. Meaning that it's highly enriched in one autocrine or paracrine interaction between different cell types, and it's very not enriched in all of the other ones. So for here with extracellular matrix organization, we see that it's very low in lots of the different cell types, and then it appears that ciliated cells are expressing a lot of the extracellular components, which interact with both ciliated cells and whatever sample group 3 was. And again, we can go down this list and find a basic summary of all of the different pathways that came up as significant and what cell types they were highly enriched for in the autocrine and paracrine signaling. So for signaling pathways in cancer, it was sample group 2 to 3. And this is a very exploratory function where you can go in and find what pathways seem to be signaling between your different cell types. And then lastly is a similar table which just summarizes the statistics using what's called an individual class importance as ranked by PyMINEr. So with this metric, we can sort based off of each of the columns. So here, sample group 0 to 0, the autocrine signaling there is most significant and important for S100 binding. And you can go down the list here and